This is lecture 4. We're going to start in on cardiovascular physiology. Now, multicellular organisms have to have a circulatory system that can move metabolic gases and nutrients and metabolic waste products around so that they're distributed throughout the body. So this is a major change in evolutionary progression from small aquatic animals that simply exchange with the external environment around them. The circulatory systems vary in multicellular organisms. Some have open systems in which the blood actually leaves the vascular system or vessels and moves through the tissues. It just percolates right through. Others have closed systems that are contained within the vessels and have a pump that moves them. They all have some sort of pump, but in closed systems it's kept within the vessels. In vertebrates like humans, the circulatory system has a pump, a heart, a closed system of blood vessels, keeping the blood separate from the interstitial fluid, the fluid between the tissues. The arterial side and, in other words, the arteries and the arterioles carry blood away from the heart, and the capillaries are where there's exchange of gases or materials, and then the veins and venules carry it back to the heart to complete the circuit going around. Through evolution, in the separation between oxygenated and oxygen reduced blood, the separation between the two has increased. In fish there are 2 chambers, and in amphibians and reptiles there are 3 chambers in the heart, and in humans, all mammals and birds and crocodilians, there are 4. So there's an increasing degree of separation between deoxygenated and fully oxygen saturated blood. In mammals, the circulation is separated, in fact, between 2 circuits. One is pulmonary, going from the heart to the lungs and then back to the heart, and then into the systemic circulation, which is delivers the oxygenated blood to the rest of the body. So what we have is a pump, the heart, and then it goes into the arterial side of the circulation. And remember that it's only at the level of the capillaries between the arterial and venous sides of the circulation that you get any exchange. Everything else are conduits for transport of the blood. The only all exchange occurs at the level of capillaries. There are, as I said, 4 chambers in the human heart, and there are valves that prevent any backflow, so you don't get any reverse flow, for example, from the ventricle back up into the atrium on either side. In terms of the size, it's about the size of a fist, and it's located on the left side, more to the left side of the chest. You have a membrane surrounding it called the pericardium. For internal organs like this, you have a double membrane, one on the wall of the chamber called the parietal membrane, and the other one on the organ itself called the visceral membrane. So you have a parietal pericardium and a visceral pericardium. The pathway of blood through the heart involves the blood in the venous side of the circulation coming back into the right atrium. So it enters into the right atrium and then passes down into the right ventricle. And the valve in between is called the tricuspid valve. And it goes from the right ventricle up to the lungs, passing out of the right ventricle through the pulmonary semilunar valve and into the pulmonary arteries. These are called arteries because they lead away from the heart. Anything that is a blood vessel taking blood away from the heart is called an artery. Any blood vessel taking it back to the heart is called a vein. It has nothing at all to do with the level of oxygenation of the blood itself. In the pulmonary circuit, that involves movement of blood from the heart up to the lungs where it gets oxygenated and then back to the heart. The vessels that carry it away are pulmonary arteries, and they're carrying oxygen depleted blood up to the lungs to get saturated with oxygen, and then the blood flows back to the heart through the pulmonary veins into the left atrium, and then it passes from the left atrium through the bicuspid valve into the left ventricle, and then from the left ventricle, while passing through the aortic semilunar valve, it gets pumped up into the aorta, and through once it goes into the aorta, it spreads through the systemic circulation. So here we see a close-up view of the heart. There's the main blood vessel bringing blood from the upper part of the body back to the right atrium. That's the superior vena cava. Here we have the inferior vena cava from the lower part of the body. They all bring blood back into this area here, which is the right atrium. Then it passes through the tricuspid valve into the right ventricle, and then through the pulmonary semilunar valve, the blood leaves. It goes into the pulmonary trunk, which splits into a left and right pulmonary arteries going to the left and the right lungs, where it gets saturated with oxygen, picks up oxygen in other words, and dumps carbon dioxide, and then returns to the left atrium through the left pulmonary veins and right pulmonary veins, and then from the left atrium down through the bicuspid valve, which is also often called the mitral valve, into the left ventricle, and from there through the aortic semilunar up into the aorta and out to the rest of the body. I do want to point out to you that you have in the walls of the ventricles, you have these extensions of the muscle tissue called papillary muscle. They're shown on both sides, and they are in fact attached to these connective tissue strands quite tough strands called chordae tendineae. So you have the papillary muscles attached to the chordae tendineae, and those in turn are attached to the leaflets of the valve. And what they do is when the ventricles contract, the papillary muscle also contracts. Muscle contraction is shortening, so it shortens and holds tightly onto pulls on the chordae tendineae, and in that way prevents the leaflets of the valve from going upwards and opening reverse. So it prevents that from happening, and in that way it prevents any blood from going back retrograde from the left ventricle or the right ventricle into the left atrium or the right atrium. So these leaflets, when they're closed, are held tightly down by these chordae tendineae. The leaflet is here, closed and held down by the chordae tendineae, these strands of connective tissue. The atria are the receiving chambers. They have on their outside there's a on the outside of the heart, lying on top of the left and the right side, is a sort of an antechamber called the auricles. So you have a right and left auricle, and they're there to handle any higher volume of, if there's a higher volume of blood returning to the heart, they can fill up as an additional receiving area. So blood, as I said, comes back to the right atrium through the superior and inferior vena cava. There's another entry point of blood returning to the heart, and that's to it receives blood from the coronary circulation. The heart muscle itself needs a lot of blood supply. It's thick, it can't get its oxygen from the blood inside the chambers, so it has its own blood vessels, and all of that blood in the coronary circulation returns through the coronary sinus into the right atrium. So you have 3 points of entry: superior and inferior vena cava and the coronary sinus. As I said, it's on the other side, on the left atrium, blood is coming back to the left atrium through the pulmonary veins coming from the lungs. And here you see that. You see some other details on the surface. You see here it says left atrium here, but it's actually the chamber inside. On top of that is the left auricle. And here it says right atrium, but that's deeper in. What you see on top, that ear-like flap, is really a antechamber, and that's the right auricle on this side here. So all of this is the right auricle, all of this is the left auricle. You see some of the cardiac circulatory blood vessels in this diagram. There's the right coronary artery and the left coronary artery. They sort of arch over the front of the heart from behind. They originate at the base of the aorta, and oxygenated blood enters into them, and then they arch over the heart and split into smaller arteries that then dive down into the wall of the musculature of the heart and go into smaller blood vessels and then capillaries, and then come back as coronary veins. So blood leaves the heart through the ventricles. They're discharging chambers. The papillary muscles I already described to you. The rest I've already described. No, all of this has been described to you before. You can read it. It's nothing different. The only thing I didn't mention is that the aorta, as it leaves the heart, it arches around towards the left side, and the blood is, coming out through that from the heart, and it arches around, and then it goes down into the abdomen through the abdominal aorta, travels down, and all along the aorta you have branches branching off, delivering oxygen saturated blood to different areas. You have 3 main branches coming off the top of the heart. The first is the brachiocephalic artery, the second is the left common carotid artery, and the third is the left subclavian. This branch here, the first one, this brachiocephalic, also soon above that divides into 2, one being the right common carotid and the left and the right subclavian artery. So this goes up and this curves around. You have here the left subclavian, left common carotid, right common carotid, right subclavian, and the brachiocephalic is just a common point from which the there's a blood vessel leading up the brachiocephalic artery in humans. Other animals are a little bit slightly different from that. And here you see them: brachiocephalic, left common carotid, left subclavian. Okay, the posterior side of the heart, I would point out to you the coronary sinus. This is where the venous return coming from the coronary circulation, in other words, blood vessels that oxygenate or deliver blood to the coronary muscle of the heart. That's where the venous return goes to this area here, the coronary sinus, and then empties into the right atrium of the heart. Heart valves ensure unidirectional blood flow through the heart. They prevent blood from moving in the wrong direction. The tricuspid and the bicuspid are valves, are also called atrioventricular valves. They lie between the atria and the ventricles, the bicuspid on the right, and the tricuspid on the right and the bicuspid on the left side. They prevent backflow from the ventricles back up into the atria, and the valves are held down by, as I told you, the chordae tendineae attached to papillary muscles, which are extensions of the wall of the ventricles. And here you again clearly see them, the chordae tendineae attached to the valve leaflets. The semilunar valves, and that includes the pulmonary semilunar and the aortic semilunar, prevent backflow of blood back from the pulmonary trunk or aorta back into the ventricles. The aortic semilunar leads from the left ventricle into the aorta, and the pulmonary semilunar valve is the point of exit of blood into the pulmonary trunk. So it leads from the right, it's at the junction of the right ventricle and the pulmonary trunk. And this just shows that if you cut at just the right angle through the heart, because the four chambers are not equal in size, but if you cut at the right angle, you could see all four valves. The tricuspid, the bicuspid, which is also called the mitral, and the two atrioventricular valves, the aortic semilunar and the pulmonary semilunar. This is a fairly nice photograph of an empty heart chamber where you see, and they put a light into it. I'm going to guess it's, I don't know, the right side, could be the left side of the heart. I think it's the right side of the heart. You have the papillary muscle attached through the chordae tendineae to the tricuspid. That means it's on the right side of the heart. Okay. So let's just look at the function of these valves, the AV valves, atrioventricular valves. Blood returns to the heart. This is essentially the cardiac cycle. We're following through the cardiac cycle as it goes through each step in a single relaxation contraction cycle. Blood comes back to the heart and fills the atria and spills into the atrium, and then the valves, the AV valves are open, and the ventricles also fill, and the valves are just hanging loosely down, and the atria is filling and the ventricles are filling, and then the atria contract, forcing whatever blood's in those atria down into the ventricles. That's what's happening here. And then following that, the ventricles contract. There's a pause of about 0.1 seconds between contraction of the atria across the top here, the atria contracting. There's a 0.1 second delay between the atria contracting and the ventricles contracting. So first the atria contract, and then the ventricles contract, and the contraction of the ventricles causes the AV valves to close, and they're prevented, and they're held closed, and no blood can go back upwards that way because of the papillary muscles and the chordae tendineae. In the next stage, which in fact what you have is contraction of the ventricles, and it's no blood is leaving, it's simply building up in pressure until it hits a pressure that causes the that causes the semilunar valves to open, and blood rushes out up into the pulmonary trunk on the right side and into the aorta on the left. And then, when blood flows upwards into a vessel like the pulmonary trunk or the aorta, there is a bit of a back wave, pressure wave. When it hits the walls, there is a little bit of a pressure wave going backwards, and that actually causes these semilunar valves to snap shut because they catch that wave and close. So the valves open and blood's rushing upwards and out. There's a little pressure wave, backward pressure wave that pushes back and closes, snaps the valves shut. That way you don't get any blood coming back into the ventricles. If we look at the microscopic appearance of the tissue itself, of the cardiac muscle, we have, this is a histological photomicrograph of the cardiac muscle tissue. And what you have are individual cells, and whenever you look at a histological slide, you should always look for, just to get a sense of the magnification, look for nuclei. And in this case, you can clearly see there's one nucleus and there's another nucleus. This is, of course, this tissue is three-dimensional, so, there are some nuclei that are hidden. They're sort of deeper down, or they were cut away higher up. So there are other nuclei here. There's more than one cell here, but there's two nuclei that are obvious. This cell here shows all the characteristic features of a cardiac muscle cell. You have multiple branches of that cardiac muscle cell. It's a single cell. It is striped or striated. It's a relatively short cell. It has multiple branches, and at the ends of the branches you have structures called intercalated discs. This here, I'm going to draw a line or arrow at it. There's several. That line there is an intercalated disc. This line here is an intercalated disc. This one here, for example, here is an intercalated disc. These are the meeting points of branches of two different cardiac muscle cells. So the cells are, it's unicellular, branched, striated, branched. They have intercalated discs at the ends of the branches, and what you cannot see are that within the intercalated discs you have many gap junctions. There are a lot of mitochondria, but you can't see them. It's not surprising that the cardiac muscle cells would have a lot of mitochondria, because after all, they are working 24/7, 365 a year, 365 days a year, so it needs a lot of energy constantly. They have to have many large mitochondria to generate energy, and they of course need a lot of oxygen because they're carrying out aerobic respiration. Right? So the intercalated discs not only anchor the cardiac cells together, but they allow, because of the gap junctions, they allow free passage of ions from one cardiac muscle cell to the next. Because of that very open kind of passage, ability for ions or electrolytes to pass from one cell to the next, the whole atria together, both the right and the left, and the two ventricles, the right and the left ventricles, each behave functionally behave as a syncytium. A syncytium is a single fused cell. So they're not all fused together, but they are functionally acting together. The atria will contract as one, followed after a short delay of 0.1 seconds by the ventricles contracting as one. You have to have that both atria contracting as one, all the cells in it contracting together to cause the ejection of whatever blood's in the atria down into the ventricles, and then soon after that the ventricles all contracting as one, all the cells in it contracting as one to shoot blood out of the heart. And here you see a diagram showing that structure, the intercalated disc here with many gap junctions in it. So this is a blow up of a cell with a couple of branches, a couple of cells, each having a nucleus. I don't know why they didn't draw this properly. There should be a good separation right here. Let me draw it because they didn't do it. I don't know why. There should be a separation there between those two. Anyhow, these are two cells with at the ends they have intercalated discs and lots of gap junctions in them for passage of ions. Now, in terms of contraction of the muscle, it is stimulated by nerves, external nerve innervation. It is a involuntary type of muscle. You don't consciously, voluntarily decide to contract your cardiac muscle. It's involuntary, unconsciously controlled. It's therefore controlled by motor output coming from the autonomic nervous system, which I've described to you. And remember, there are two divisions. There's the sympathetic. I'm just going to write SYM for the sympathetic, and the parasympathetic. And these have opposing kinds of actions on the tissue. They also, however, unlike anything else in the body, they have their own type of stimulation that's built in. So it's called automaticity or autorhythmicity. These, in other words, they have tissue that's self-excitable. And essentially what that means is they have their own pacemaker. The pace of the heart is normally established by the heart's own self-excitable tissue or pacemaker tissue. And that is why it contracts in the manner it does, and I'll go through that. This autorhythmic property is in specialized autorhythmic cells that form part of the intrinsic conduction system within the heart. These autorhythmic cells, and I'll describe the anatomy of that system in the next slide or two, they initiate what are called action potentials. An action potential is a sudden reversal from negative charge, intracellular negative charge, to a more positive charge. So what happens is that you have these autorhythmic cells generating these action potentials, sudden switch from more negative to more positive. All what's called excitable tissue in the body has this ability to generate action potentials. However, autorhythmic tissue is different from any other excitable tissue. Excitable tissue includes any type of muscle or nerve cell, neuron. So muscle cells and neurons or nerve cells are excitable. They can generate action potentials going from an internal negative charge to the more positive. But for every tissue other than these autorhythmic cells, they are stable until they receive a signal, and it's only when they receive a signal that they, if this is the voltage inside the cell and this is more positive and this is more negative, that is, they will stay there at the resting membrane potential until they get a signal. So they will, if they do get a signal, then they will start what's called depolarizing, moving from the more negative towards the more positive. I'm going to change this. This is moving from a more negative charge towards the more positive if they receive a signal, and the signal usually is some sort of neurotransmitter signal. And when they hit this dotted line, this level called the threshold, then it generates what's called an action potential, depolarizes very quickly, goes quite positive, then repolarizes, comes down, and then goes back to rest until some other time when it receives the appropriate signal. That's what skeletal muscle does, that's what smooth muscle does, that's what neurons do, and that's what cardiac muscle cells do, but not cardiac autorhythmic cells. They do something quite different. They don't need a signal to come in to generate an action potential, a sudden change in their internal charge from negative to positive. They are constantly depolarizing. So they're not staying down at a resting membrane potential. They're depolarizing. They hit the threshold and you get an action potential. And then it reverses and it comes back down. And then again, they depolarize, then hit the threshold, and again. And that's why at a certain pace, and that's why they're called pacemaker cells or pacemaker tissue. And that's why they say that's why it's described as having its own pacemaker, its own autorhythmicity, because of this specialized tissue that triggers a action potential. The action potential generated by the autorhythmic cells will trigger depolarizations in the other cardiac muscle, the normal contractile cardiac muscle. And here you see that. So these are internal recordings using these microelectrodes that are put into cells within the pacemaker tissue, the specialized tissue that's located in the heart in a specific spot. I'll describe that later. This is pacemaker tissue that generates spontaneous action potentials. There is no resting membrane potential. It doesn't sit there and wait for a signal before it depolarizes. It spontaneously depolarizes till it hits that threshold level. So here along the vertical axis we have the internal voltage inside the cell, and across the horizontal axis we have time in milliseconds. And so because of the activity, well, I'll explain it in a minute, you get a constant steady slope upwards of depolarization until it hits the threshold, generating what's called an action potential, a very quick change upwards, positive, and then it repolarizes, comes down, and then it starts again. Slow depolarization, that's called the pacemaker potential. And when it hits the threshold, again, another action potential, and then it comes down, repolarizing, then depolarizing, etc., etc. These changes in internal voltage or charge inside the cell are due to electrolyte channels that are opening and closing. The electrolyte or ion channels in the specialized pacemaker tissue are present, and they go through the following changes I'll describe to you. I want to point out to you that these channels for ions or electrolytes are specialized, so you have separate channels for potassium, separate channels for calcium, separate channels for sodium, right? Now, I also would remind you, and you should have learned this before, but if not, anyhow, I'll explain it to you. All cells have pumps. Pumps aren't channels. Channels are, you know, it's like a tube that opens. It's like a gateway that opens and lets a particular ion out or in. But pumps are physically pumping ions, and every cell has sodium potassium pumps operating. And the way these pumps work is that sodium gets pumped out and potassium gets pumped in. So the sodium potassium pump is pumping sodium from inside the cell. I'll put an I here and an O here, from inside to outside, and potassium from outside to inside. Okay? Well, if you have a positive ion going out of the cell from the inside to the outside of the cell, then the inside of the cell will become more negative. If you have a positive ion going from the outside of the cell in, then the inside of the cell becomes more negative, right? I'm sorry. If a positive ion goes from inside out of the cell, then the inside of the cell becomes more negative, and if a positive ion goes from outside in, then the inside of the cell becomes more positive. All right. Well, in terms of channels, there is, in fact, there is a low potassium permeability. So potassium, and remember, an important point here is because of the sodium-potassium pump activity, sodium is higher in concentration outside the cell because it's getting pumped out. So the sodium concentration outside is higher than inside, and the potassium concentration is higher inside than outside. If a channel opens for potassium, then potassium is going to rush out because movement of ions is dependent on the concentration gradient. So where potassium is higher inside, if a channel opens, it's going to move out. This is the sodium-potassium pump action here. But if a channel opens for potassium or sodium, then they'll move in the direction of from high concentration to lower concentration. At this point here, we have a low potassium permeability. In other words, potassium channels are not open. But you do have leaking inwards of sodium. Okay? There is some permeability for sodium, so sodium leaks into the cell. There is some opening of sodium channels, and you get slow entry of sodium in, so the voltage, the membrane potential or internal intracellular voltage is slowly moving, and slow is relative, this is milliseconds, slowly moving towards the more positive because of that slow entry of sodium in. Once it hits threshold, then what you get are calcium channels opening, fast calcium channels open. I should point out to you that in terms of, I'm going to list them here on the side, in terms of concentrations, sodium, because of the sodium-potassium pump, has a higher concentration outside. Okay, so I'm going to draw it this way. This is my own system. Higher concentration outside. If a channel opened, it would go inwards. Remember, it's positively charged. Let's see, let's change this. So, sodium is higher outside. Potassium is higher inside. And calcium is higher outside. Okay? So if you have slow sodium entries, sodium is leaking in, the membrane potential or the charge inside the intracellular charge is going from moving upwards in this direction, more positive. When it hits the threshold, you get opening of fast calcium channels. In other words, calcium permeability just went up because the calcium channels opened. And calcium is higher outside, so the flow is from outside in. And calcium is a positively charged ion, so the inside of the cell quickly depolarizes during this action potential. It's called an action potential, and it goes very rapidly up, more positive. And then calcium channels shut. So calcium permeability drops off. But potassium permeability rises. Potassium channels open. Remember, potassium is higher inside than outside. And so potassium will go out, and the inside of the cell becomes more negative. It repolarizes. It goes down towards a more negative. And then the whole process starts again. You get low potassium permeability accompanied by some slow sodium entry, and again, depolarization to the threshold. And that is how these impulses are being generated spontaneously in the pacemaker tissue of the heart. And these will spread. Each time the action potential is generated, it spreads into the cardiac muscle cells that have contractile ability, the vast majority of the rest of the tissue of the heart. The actual pacemaker tissue is located in what's called the sinoatrial wall, sinoatrial node in the wall of the right atrium. And it spontaneously depolarizes, and that depolarizing impulse spreads down to what is called the atrioventricular node, or AV node. So it's initiated or generated spontaneously in the SA node, spreads down to the AV node. There's a slight delay of 0.1 seconds, and then it goes from that AV node down through the bundle of His and into the bundle branches, and the bundle branches lead to the Purkinje fibers that spread all over the wall of the ventricles from below arching upwards. So you get this depolarizing wave going through this what's it called? Intrinsic pathway, intrinsic excitable tissue in the heart, starting at the SA node, going to the AV node, going into the bundle of His and the bundle branches and the Purkinje fibers. When that SA node initiates an action potential, it also spreads all across the atria into the atrial cardiac muscle cells, triggering. Remember, muscle is excitable tissue. It can contract. It can get an action potential and contract. It gets depolarizes. It depolarizes because of this signal coming from the SA node, and you get contraction. So you get a wave of contraction instantly across both atria. Remember, they are functionally acting as one. They are a functional syncytium. They will contract as one. And then it spreads, it's also spreading to the AV node and then down into the ventricles with a delay. So first the atria contract, and then the wave spreads across the cardiac muscle cells in the ventricles, and the ventricles contract. The impulses are generated about, from the SA node about roughly, there's a range, but it's say 60 to 75 times per minute. And the delay at the AV node is 0.1 seconds. This just describes the pathway which I've talked about before. So first atrial contraction because of the spread of the wave across the atria, and then ventricular followed by ventricular contraction because of the delay at the AV node and the spread of the wave, depolarizing wave across the ventricles. So that's the internal intrinsic stimulatory system in the heart, the pacemaker tissue, but it also can be affected by external nerve innervation. And I'm going to stop here. This is the end of lecture, lecture 4, part 1.